Now it's my distinct pleasure to welcome Rodney Dietert, who's from Cornell University, who's going to talk to us about the microbiome and immune system in environmental risk of infectious diseases. So please, welcome. Thanks, Rodney. And that's just, this is your advance, and that's just, that'll go to tell you when your time's fantastic.
Thank you so much. Well, I'd like to start by thanking the organizers for the opportunity to present and to be a part of this exciting and important forum. Earth is a microbial planet, and we heard bacteriophages being predominant, viruses, but the other microbes as well. And I hope to present a picture that will basically say we can have win-win between our microbial components, those of animals and Earth's, or lose-lose. And so a lot of it is really the integration between the environment of Earth, the environmental risk factors, and the health of all biota on Earth and how they interplay. I'd also like to thank my primary editor and wife, Janice Dietert. Pretty much everything I write or present goes through her first, thank goodness. This is my disclosure. So in addition to being at Cornell and 41 plus years as a faculty member, as of last week, I'm actually emeritus, now professor emeritus. And I also have a connection, a consultancy with a 2018 startup company, Seed Health, and in addition, a publisher. So my disclosures. So I want to start with talking about a few dogmas from the 20th century that are either destroyed or I would advocate should be destroyed soon, and the ramifications of that. And so as I build the talk, it's going to play off of the destruction of these dogmas that we used to embrace. Some of them I probably used to teach back in the 70s at Cornell. The first is an immunological one, that the newborn is fully prepared immunologically to fight all diseases. I was taught, and I used to teach, that the newborn baby had everything the baby needed from the immune system. We could count all the cells that we knew in the 70s were there. Everything was good to go. And that's not true. Baby comes out of an environment that's been protected. It's protected from the mother responding against the paternal antigens that the fetus carries and not rejecting the fetus. And that environment, we would call it Th2 biased, allows some aspects of the immune system to sort of mature at a faster rate than other aspects of the fetal immune system. There are other things going on with the innate immune cells, and such that the baby is really born in a state that is biased a bit towards some types of responses, and the others have to catch up. And we learned that they catch up, particularly if you grew up on a German animal farm, and the famous German barnyard studies that led to the hygiene hypothesis said, wow, as a pregnant woman or as a newborn baby, if you're out with animals and you're on a farm that doesn't use pesticides, that's actually good. You have a lower risk of asthma and other things, and immunologically you can measure that. And so the baby has to catch up. And that catch up, I'm going to argue, is largely requiring the microbiome. And the microbiome has to be robust and in place and match that baby's host genome fairly well for success. And that'll be part of the talk. The other, number two, so the baby's not good to go. The baby has to mature postnatally, immunologically, or the baby's open for infectious diseases and eventually inflammatory-driven host damage to organs, which would be the NCDs. Number two, species barriers are almost exclusively chromosomal gene-driven. And certainly they are largely chromosomal gene-driven. But there are some exceptional examples that now show that the interaction of the microbiome and the immune system really define who we are, our tribe, and our species in some cases. And that's a fairly new finding. Number three, that most microbes are a threat to human health. And this was an unfortunate development during the 20th century, whereas we now realize that most microbes, and at least some microbes, we need to be healthy and to thrive. And that is a sea change. So it means managing microbes becomes important. And that'll be another theme. And finally, that the human mammal, as a toxicologist and former director of Cornell's Institute for Comparative Environmental Toxicology, as a toxicologist, I did a lot of work on safety targeted toward the human, thinking the human mammal was the end game, but it's not. We're a superorganism, we're a walking coral reef, and so it is really a different target in environmental health, safety, and risk assessment, and that is for our thousands of species that are a part of us. So this simply illustrates the gene and metabolism game of the superorganism, and that is that, of course, the Human Genome Study had many laudable goals, expectations, achieving some of them, but nevertheless, the number of genes on the chromosomes that we carry is somewhere between 22 to 25,000, a couple thousand more than the earthworm, so yay, we have more than the earthworm. But individually, we have about 3.3 million microbial genes in our body, and across humans, just under 10 million total. So we're more than 99% genetically microbial, and we have a slight majority, about 57% of our cells are microbial. It used to be thought it was 90, but it's closer to half and half. So by a lot of measures, we're majority microbial. And those 3.3 million microbial genes are making enzymes and cell signaling molecules and doing stuff, just like our chromosomal genes. And if you'll notice, one of the great liver metabolism enzymologist, Kurt Clausen, former SOT president, his most recent papers are actually on microbiome metabolism. And there's a reason why. Because there's tremendous potential both in direct metabolism by the microbiome and by epigenetically regulating what goes on in inducible enzyme systems in the liver and elsewhere. So they are doing a lot and they're controlling a lot. And that's driving a lot of the change of thinking. So one of the things that I think is really useful to recognize, particularly at this session where we're integrating environmental health, infectious agents, host defense, is that the toxicological routes of exposure that I've lived with for decades and embrace and memorize and think about in my sleep, the routes of inhalation, the airways, oral, drinking water contamination, Flint, Michigan, lead, dermal, and to some extent, lesser but urogenital also, for toxicological. Those overlap, are coincidental with, and I'm in the microimmunology department at Cornell, portals of entry for infectious agents. And that would be influenza in the airways, right? Breaks in skin for infections. Ingestion of all kinds of wonderful things, through water contamination and food, right? Some of the lettuce, fecal deposits on lettuce we just heard about. And urogenital, they're the same. And where's the microbiome located? And that's shown on the right panel. And that is where we're exposed to the external world, the external environment. And that would be the airways, the GI tract starting with the mouth, airways starting with the nose, skin, and urogenital. And then there's a microbiome in breast tissue and some other locations in addition to breast milk as well. These are the same. So we're really talking about a few body sites where that determine what we are and where we interface with the environment, whether it's chemicals, drugs, food, or pathogens. And obviously, I would say a lot of attention needs to be given to those interfaces, and that'll be part of the theme as well, is that's where the action is. Now, immunologically, that's wildly different, as I'll show you in a minute, from what I learned and what we used to teach. So how important are the two topics that I have in my title, the microbiome and the immune system? And I would argue they define who we are, they define the tribe we're in, and what we are in some cases. And the examples here are shown from the Bordenstein lab at Vanderbilt and their colleagues, and from Wang et al., a collection of European, mainly German, institute researchers, showing two examples that change the ballgame in terms of species barriers being being almost exclusively chromosomal gene defined. And what these investigators show is that first in the parasitic wasp, and that's a generic wasp, that's not Nasonia, and then the house mouse is on the left. In 4 species of parasitic wasps, they are different species because the larval stage does not survive. The larval stage, if you try to hybridize those wasps, is destroyed by the immune system in a massive inflammatory reaction, kills it. But they don't have to be different species. If you treat with antibiotics and remove some of those microbes, you can mate them, and the hybrids grow up. They're fine. They're viable, they reproduce. The microbe-immune interaction makes it a species barrier because the immune system has grown up long enough that it will not tolerate those microbes in the hybrid. Now, there's the same story in Europe. 2 subspecies of house mouse. House mouse that are geographically defined, so pretty much Eastern Europe. Again, I didn't have the copyright permission on the map. I'm not showing you the map, but Eastern Europe, Western Europe, geographical separation in this case. The hybrids you can produce, but they don't survive long, they don't thrive. Why? Because of massive inflammatory reaction. Immune system rejects it. It's like not that different than a horrific combination of non-communicable diseases. And yet, if you treat, again, with antibiotics and create something closer to a germ-free mouse hybrid, just fine. No subspecies barrier, no problem. So that tells you that interface of what the immune system sees and the microbiome is absolutely sacred biologically to the extent that it can separate or bring together what we thought was a species barrier, cross-species barrier. Pretty important biologically. So from there, I wanted to comment about the idea that the newborn's immune system is absolutely good to go, going to fight all diseases, just great, as the fallacy or the dogma that needs to be retired. We know that environmental exposures and adaptation are required for full immune maturation. I already mentioned that the newborn comes out with a predisposition for an inflammatory reaction trigger in some cases, but there are other parts of the innate immune system that still has to mature. So there has to be brought into balance on the inflammatory reactions that are going to occur, and the Th1 antiviral type responses, anti-cancer responses have to really start from ground zero and be brought up in combination with the Th2 bias that already exists. And that's just dealing with some fundamentals about the immune system. As I'll show you in a minute, you have to essentially recruit and rebalance the immune cells as they exist in the periphery, and that would be where it interacts with the microbiome and in all of our tissues. Microglia in the gut, Kupffer cells in the liver, dendritic cells in the skin. All of these cells that are being housed as residents. So gut microbes at microbial surfaces drive the newborn's immune development. And there is an interchange between the immune system and the microbes that are there, IgA going across in one direction and short-chain fatty acids and other signals going across from the microbes to the immune system. And that is absolutely determining what happens. So if I used to teach that the thymus and the bone marrow were where the action was for the immune system in the 1970s at Cornell, we now know that 2/3 of the immune cells are in the gut. So guess where you want to be looking when you're really looking at health risks, I believe, and particularly postnatally, and that's at these barriers in the gut, in the airways, in the urogenital tract, and looking at what's going on. So some aspects of inflammation have to be ramped down. Other things have to change. I listed one of the signaling. It's an interferon regulator there that's a transcriptional factor. That's one of the things that has to be changed to protect against certain kinds of infections. There are helper and regulatory populations among T cells that have to be changed, particularly in the periphery. B cell, natural killer cell, dendritic cell maturation, particularly in their first 3 months postnatal, are critical. You don't want to miss these windows. These are developmental windows. Doesn't mean you can't adjust the immune system when you're 60, in your 60s like me, but it's a much tougher task than protecting early. And we know from Developmental Origins of Adult Health and Disease, DOHA, the journal, and that NIEHS and the NTP have supported so well and been a part of, that's real, that you can do so much earlier that's much more effective and permanent than putting out fires later. And there are specialized immune cell populations, so the microglia in the brain, invariant NKT cells, and things like this in the periphery that literally the size of those populations, the maturation activation stages in the newborn and in the early infant are absolutely critical, both for resistance to infectious diseases, but also pathology in tissues. So I want to just mention a couple of windows. Postnatal windows, this is from rodent literature, but again, really good reason to believe that these things are going on in humans, these parallels. So one of them is from the Harvard group, Blumberg's group, where they've basically shown that in some of the inbred strains of mice, there is a population in the gut called invariant NKT cells. You need them there. If you don't have them, it's a problem, but you need them in the right amount. So between birth and 1 week in a mouse, in these strains, these will have a proliferative burst. Bacteroides fragilis produces a sphingolipid that dampens that burst, makes it about half the size it would be otherwise. If that sphingolipid is present, the burst is dampened, the mouse is protected against autoimmune inflammatory bowel-like diseases, colitis, which will happen later in life with an environmental trigger, by the way. It's just sitting there. The immune system's dysfunctional. You don't know it till you get the challenge. Could be an infectious agent challenge, could be a chemical challenge, drug. So in this case, it's not facilitating something to happen. It's keeping it from happening in excess, where it otherwise would. Got to have the bacteria there. Got to have the metabolite there. In another case, there's FOXP3 positive T regulatory cells, the natural Tregs, they're called, and they've got to be recruited into the skin and interact with the commensals so that you have tolerance throughout life. And again, there's a narrow window for that. So in both of these cases, you miss that window, you can put that sphingolipid in the gut all day in mice after 1 week of age, and those animals are still set up for inflammatory bowel. You missed it. Early co-maturation, critical. So I've tried to, published in Reproductive Toxicology, the idea of this as a systems biology unit, because these specialized immune cells interacting across in the gut, a one cell thick barrier with the commensal mutualistic microbes and pathogens among them as well, or potential pathogens, is absolutely critical. And the signaling, the output, the exposures there is really something that we should know a lot about. And so I put a name on it, and I don't really care whether people use the name or not for the unit. I'm more interested that they focus on that together as being important in our interaction with the external world. And keep in mind, this is the outside of the gut we're talking about. It's exposed, right? And dentists will tell you they can look at oral hygiene and tell you a whole lot about what your immune microbiome health really is in the end. So when these microbes that are on us and in us get exposed to chemicals, drugs, and food, what do? They can sequester it. It may be something they want, a metal they need. They can avoid it or exclude it, keep it out, and they have systems for doing that. They can metabolize it. You're going to hear a wonderful talk on arsenic later. I'm going to use arsenic just briefly to make a couple points later on. So they can metabolize it depending on what metabolic products you get. That's going to influence your physiology, sometimes big time. They can signal among themselves, among interbacterial as well, and to the host. They can die. And if it's a keystone bacterium with a unique function you've got to have, that's bad. So food emulsifiers kill Akkermansia, which maintains our mucin layer. We thought they were safe. They were never tested for microbial safety. That's not good. because there's only 2 species that help regulate mucin layer in the gut. So depending on what dies, that could be bad. They can expand. That may be good, may be bad, depending on what's expanding. And clearly bacteria that can use a component of food as an energy source, they're going to grow. Others that can't use it, not so much. Or they can translocate. And microbes moving across niches is usually a really bad thing. Usually leads to pathology. So you can think about our veterinary students where I teach at Cornell have a case where bacteria, urinary tract blockage leads to a tear in the peritoneal cavity. They get into the peritoneal cavity. That's not a good translocation. It's a very bad thing. Septic shock happens. So that's usually bad. You want them where they're supposed to be, the niche they're attuned to. And they metabolize in a variety of ways. And again, this is from the Clausen and colleagues review. So they can increase or decrease drug availability. And a lot of drugs, cancer therapeutics, digoxin, a whole host of drugs require microbial metabolism for active components. And depending on what a patient has individually, digoxin can be effective, for example, ineffective, or toxic and kill the patient. One bacterium determines that. And I think we want to know how much bacteria are in those patients before we give that drug as a physician. But it can also rescue drugs, make them more effective, and reduce side effects, knowing this. So direct metabolism of drugs, it can inhibit detoxification, or it can biotransform food components. They can generate AHR agonist. They can convert a prodrug into an active drug. They can respond to one drug by inactivating host enzymes for an unrelated drug. That's very insidious. And they can regulate host metabolism overall, HPA axis, liver. So managing these microbes would be a very useful goal. Okay, we in the US have had our microbiome destroyed to about 35 to 40% as a population. This is based on Gloria Dominguez-Bella's work comparing against Amazonian indigenous populations. That has ramifications. They can be like us in a disease spectrum and susceptibility by moving to the suburbs of Sao Paulo, starting to eat that diet, and then moving into Sao Paulo, which is, I think, urban areas have been termed a microbial wasteland. So we may think they're green, but they're not microbial rich. And that they can destroy the microbiome like ours has been damaged. So there are other things that we're doing that contribute to this. You hear about antibiotics, but that's the tip of the iceberg. Keep in mind, drug companies have now been surveying existing drugs. If they were not screened for microbial safety, then you should not assume that they're safe for the microbiome. So electrocesarean is one of the things that clearly, if the baby's not seeded with a robust microbiome, the baby will acquire microbes from places like hospitals. And it may not be what's desirable. Prophylactic use of antibiotics in animal feed absolutely has been a disaster. I even had an editorial in the Christian Science Monitor back in the 90s railing against it because they're used as growth promoters. They're not a health strategy. They're simply to have larger chicken breasts per pound feed that goes in. Because if you reduce the inflammatory TNF-alpha and IL-1, you don't get muscle catabolism, so your chicken breasts are bigger per pound in. It's a very bad reason to contaminate the world with antibiotics. But you look at that and you can see the ramifications for antibiotic-resistant genes, the transfer, the zoonosis as well to humans. You look at captivity, and this is interesting. I'll ask you to consider this in a minute relative to humans. So howler monkeys eat 57 species of plants in Vietnam, 1 species of plants in American zoos. So what happens to their microbiome? It gets degraded. They don't have as rich a food source contributing to growth of their microbes in the gut. So where do they get their microbes? They get them from zoo handlers. They become humanized. And by the way, our lab animals from Eric Erickson's study at University of Missouri, our lab animals acquire lab technician microbes because they don't actually have as robust a microbiome as wild mice do. That's a pretty limited environment. It's like the urban equivalent for them of megacities for us. and they actually start to be humanized. So you switch animal handlers or move your rats or mice to another facility, good luck. You've changed the microbiome. So Ellen Silbergeld and I published a paper in Toxsci 3 years ago now, and basically argued that this is the NRC developed model that served us really well since 1987. Ellen was on that panel that helped develop this. And it's where we go for trying to predict an external exposure, an exposome, through internal biological effective dose, altered structure, function, and eventually clinical disease. So you can determine what level is safe, what is not. But Ellen and I looked at this and said, and this was part of a conference actually on metals and infectious diseases that deteriorated into a microbiome conference. The reason being, if you don't know how our microbiota handle metals, you don't know what's going on into our physiological systems. Whether it's selenium or arsenic or zinc or lead, you don't know. So that was actually the turning point where we thought, there's a problem. So we basically, it's like how to make a brilliant new figure, stick the microbiome in. So yeah, exactly, you can see, radical change. But it is a radical change in a sense, because the microbiome is our gateway, it's our filter. We see the environment through the microbiome. It's at all the external portals, right? So every chemical, drug, food additive, infectious agent has to come through there, in a sense, or come out of there for some of the embedded pathogens. So relative to body dose and anything downstream of that, you don't know until you know how the microbiome handled it. And of course, there's some individual variations, some interesting things that, and it's malleable. You can change a lot of those 3.3 million genes. Easier than using CRISPR on chromosomes, So what we basically said is, wow, we've got some bad news, good news. Bad news is, as toxicologists, we don't know what's safe. Good news is we're going to be working for decades because we get to do it all over again with the microbiome. But in a sense, it's real. In a sense, it's real because we're seeing that now with drug safety and chemical effects and different individual variation. So you have personalized risk assessment that goes down to starting with this. So that leads me to the final two topics I want to bring up, colonization resistance, and then I'll use the arsenic example. So colonization resistance, I think, is really important when you consider infectious agents and risks. Not that the immune system isn't, but you've got to give the immune system a break and start to use microbes against microbes to protect human health and protect the health of animals, in fact, the whole biota. So you've heard of the old term, colonization, competitive exclusion, which was used back more in the 70s and 80s, and Nurmi Concept, the Finnish researcher. This was actually applied. I was hired in the poultry science department at Cornell in '77, right about the time we had a Salmonella E. coli outbreak that then became a zoonotic threat. People stopped eating eggs. So what did they do? How did they address that? Because it was being spread through the oviduct vertically, and it was being spread horizontally, chicken to chicken, Salmonella. So they loaded the birds up with Lactobacillus acidophilus, and it solved the problem. And the product's been on the market, and these people make pennies on the egg, believe me. It's a small profit margin. They don't pay for something unless it's going to pay off. And that strategy, competitive exclusion, colonization resistance, has been in place. It's the good news part of animal agriculture where you actually can protect against pathogens in the species and that are zoonotic by not allowing the pathogen to gain a foothold, in this case, Salmonella and E. coli. So the more specific term is colonization resistance, which deals with all of the interactions, not just the idea of a niche physical exclusion type of approach. And this works by the categories I've listed. Nutrient limitation, secondary bile salts are really important. So bile salt metabolism by the microbes is important. And Curtis Clausen talks about this as well. Short-chain fatty acid production like butyrate are really important. There are antimicrobials that some bacteria make that block other bacteria. And cell-cell contact. So there's the type 6 secretion system and LGX proteins that are dependent. They're a type of toxin family that some bacteria have, and they can help to restrict pathogens very specifically. And we know about these, and we can use that to ensure that babies, infants, and us in older age actually have that frontline screen that's going to maximize resistance to major infectious agents. And that will also help protect the immune system in the lining. Now, one of the ways this works, again, is to keep microbes you do not want against that epithelial layer away from it. And there are two different thicknesses of mucin. I mentioned there are only a couple of bacteria species that regulate mucin production. You don't want to damage those or lose those, or you are in deep trouble, both for infectious disease and NCDs. So essentially maintaining those and keeping the immune system communicating in a nice regulated manner is useful. But if those pathogens are able to reach the epithelial layer, send LPS, lipopolysaccharide, through activated macrophages and the innate immune cells and produce inflammatory responses that further damage the epithelial barrier and cause other leakage of other things, then you're on your way to several categories of different elevated disease risks, including things like metabolic syndrome. So this is one of the ways that colonization resistance works, is to maintain that system. That's one of the reasons I've argued for a systemic look at all of our barriers where we're exposed to the outside world. Well, how many pathogens does it take to do this? Is this just a heroic effort? And there's actually some really encouraging news. So this is from a study that used mice, but a human enteric pathogen, Salmonella enterica, Cerevar typhimurium, and they asked the question using metagenomic-directed approaches to package together bacterial species that they could install in a germ-free mouse. And they basically said, what do we have to put in to completely protect against this human gastrointestinal pathogen, a major pathogen? And the answer was as few as 15 bacterial species will give you the metabolic and otherwise environment to protect. But the great thing was they used a directed combination, metagenomically. So it's only one pathogen, but it's an important one. And we can do that. And we should do that, because that's also going to help the immune system, particularly if we mature the immune system correctly. And I do want to raise the interesting issue that we talk about the infectious disease, non-communicable disease barrier, and I've been one of the worst on NCDs, used that a lot. If you think about what I've told you about humanized howler monkeys and mice, and if you're depleted in your microbiome and you start to just pick up what's around, then you start to ask questions like, are non-communicable diseases completely non-communicable? Is that a hard barrier, or is that a softer barrier? Because we're really talking about installing microbes and exchanging microbes. I have dogs. Dogs are great for spreading microbes around, detecting them. They can detect the VOCs off of Clostridium, by the way, if you difficile. If you train them the right way, they're the best detectors of it. And think about how dogs greet each other. Is it face-to-face? No. They're going right after the microbial detection end of the dog, right? So a couple of things about arsenic. You're going to have a wonderful talk coming up, I know, that includes that. But I wanted to show you that this touches upon all the aspects I've mentioned already. This is why this system is so critical in every part of it. So there are sulfate-reducing colon microbiota that will thiolate, particularly methylated arsenic, and make some of the most toxic forms of arsenic. So your individual risk, if you have exposure to arsenic via drinking water, sort of a one-shot thing, your individual risk can be affected by how many of these bacteria you have in your colon. And we vary in that. And that would be a risk factor for that toxicant for that individual. So there's variation in these bacteria. Arsenic exposure can also change the gut microbial composition, and it will change microbial genes, and it depends on duration. You know, is it an acute exposure or chronic, and of course the dose. But it can damage the microbiome in a way that presents major increased risk. It also can increase the risk of certain infections, and this is epi studies from NHANES, but Hep B, liver infections go right along with the burden of arsenic following exposure. And you can show other viral infections in particular also. I mentioned it can change microbial composition, and then that will change subsequent arsenic metabolism. But the other thing is, if you have an acute, fairly low exposure, there are microbiota, not in the colon, that will actually protect. They will actually block arsenic from being able to get through the barrier and to your physiological systems. And so there are groups that protect, there are groups that make very toxic metabolites, and understanding those, and this is just one case with one toxicant, can be really important. So if it gets through the immune system, because I mentioned the immune system is important too, what's it do? Well, the bottom line, if I had to put 25 words or less, is it ages the immune system. So it reduces telomere length. It particularly targets early T cells, naive T cells. Well, those are the T cells that you lose as you age and become geriatric like me. You're restricted, you're inflexible in some of your making new responses to new things. Arsenic does that in early life. And early T cells, from Scott Burchell's group at University of New Mexico, early T cells seem to be less able to pump it out. So they get it in there and they can't get rid of it, and it becomes a problem. They're somewhat targeted. So I just wanted to show you, this is why toxicologists and developmental biologists and still have careers ahead, show you my sort of worst case scenario. I was asked to do this for a Neo Reviews article dealing with infections, to show you that the pregnant woman, and by the way, she might have something like diabetes or obesity and an associated dysfunctional microbiome as well. But you have stress, you have exposure to environmental chemicals, could be secondhand smoke, could be PAHs, particulate matter. There's F1 immune programming for reduced T cells in the infant, elevated risk of a number of diseases already, cesarean delivery. It's a surgery. You get antibiotics with that as well. Depletes the microbiome, reduces colonization resistance, higher level of risk of infection in the infant, right? Maybe there's formula feeding. You miss the microbiome part of breast milk, which is transferred just the right stage. You miss the oligosaccharides that are only able to be used by microbes. They're not there for the human mammalian part of us. And that goes on to further damage the immune system and promote inflammation. In the end, it is the misregulation of inflammation that's a huge problem. And then we have our favorite toxicants, the heavy metals, BPA, and others. So this is like, not to scare anybody, but to say, look at all the opportunities where we can do things differently and better. So I'd like to stop with that and just summarize to say the microbiome is the lens through which we see our external environment. Oh, okay, I have to read it off there, sorry. And, it's our first line of defense. And so it is malleable. We need to be using that in our strategies. The immune system has to co-mature with it. And I've already shown you that at the extreme, the immune system will kill things. It will not permit a hybrid to develop. So there's probably some matching that goes on. Think about that with fecal microbiota transplantation. You probably actually want a pretty good match for what your immune system has been used to interacting with in an endotype, in your geography and your ancestors' diet and everything that has gone into your genes that do contribute to your immune system. Optimizing colonization resistance is really a must. Let us use the first line of defense to help us resist infectious diseases. It's not the only answer. Obviously, we need a lot of other things, but we can use this. We can use this in humans. We can use this in animals. We can use it as these interactions occur. And finally, managing the microbiome immune interface across the barriers is really going to be, I think, a significant part of both preventative and therapeutic health, whether it's humans, animals, or otherwise. So thanks again. Really appreciate this.